Am I audible? Can you see the screen? Hello. Yes. Okay. Fine. Done. So the factors affecting the enzyme activity is one of the factors is temperature. As I said, each and every enzyme is optimum at particular temperature. We have discussed this part of the temperature. What do you mean by optimum temperature? The maximum activity shown at this temperature is known to be an optimum temperature. Enzymes get denatured at higher temperature. At lower temperature, they remains to be inactive. Is the point clear? At lower temperature, they remain to be inactive. At higher temperature, they denature it. And optimum temperature is the best temperature at which they shows activity. Clear all? Have you all drawn this graph? This graph is important. Can we move to the next? So which is the structure that enzymes need to be active? At which structure the enzymes are active? Primary, secondary, tertiary, quaternary? Tertiary. Very good. Tertiary, any changes that affect the tertiary structures will affect the activity of enzyme. Remember this. Any changes that affect the tertiary structure will affect the activity of the enzyme. Is this point clear? Okay. Effect of pH. Most enzymes exhibit optimum activity at pH that pH range from 5 to 9. High or low pH value than optimum value will cause denaturation of the enzyme by ionization. So write down this. Write down this. Every enzyme has an optimum pH. When it is most effective, okay, optimum pH means where the activity is high. Activity of enzyme is high. A rise or a fall. It can be a rise or a fall in pH reduces enzyme activity. Enzyme activity. Okay. Some enzymes are best at acidic medium. Some enzymes are best in an acidic medium. And some alkaline. And some in alkaline. Can you quickly quote me some examples that are best in acidic and the best in alkaline? We have done the digestion absorption chapter. So you know which some, there are few enzymes that are active at some point of the pH. Can you quickly quote me those examples? Very good. Gastric. Gastric. then that is acidic. So any alkaline you want to quote, any alkaline. Sulcus entericus, very good, Yash. Sulcus entericus, basic. Pancreatic, very good. The first enzyme, the start of the digestion happens from where? The first enzyme, the start of a digestion happens from where? Very good. It's a salivary amylase, right? Salivary amylase. It's a basic, right? See, when the bolus is coming, bolus means I hope everybody knows. After the food is chewed, the leftover saliva is around it, and this is called bolus, right? This bolus, when it reaches the stomach, it the salivary amylase will lose its activity because the stomach is acidic. The enzyme that were active in mouth, they are not active in stomach. Are you understanding the difference? The optimum pH, how the pH plays a role in activity of enzyme? Everyone? Yes. Yes or no? Okay, next. So this is an optimum pH of the effect. See, if you see here, you can see the slide. pH affects the formation of hydrogen bonds and sulfur bridges in protein and so affects the shape. So you have pepsin. Pepsin is acidic, active in acid. Trypsin, where it is active in around 6 to 8, and there is arginase that is active. If the same enzyme pepsin is present in acidic media, in basin, if it's here, pepsin is here. If it's present somewhere here, the activity is destroyed. They remains to be inactive. So trypsin can be active between the pH 3 to 6 to 8 itself. They do not show activity in acidic media. Please do draw this graph. Any doubts?
So, ma'am, please show the previous slide.
Previous slide. Okay. Can you see the previous slide?
Am I
No,
recording?
ma'am.
Yes, I'm recording.
Ma'am, it's not visible.
It's not visible. Can you see it now? Is it visible now?
Done.
Okay. Next. Substrate concentration. Next, we'll be studying the substrate concentration. You all know the substrate bind with enzyme. So when a substrate concentration is increased, the rate of enzymatic reaction also increase first. Because enzyme is present, substrate is present. Initially, when you add enzyme along with the substrate, the rate of reaction increases initially. When the reaction rate reaches to the Vmax, for example, if this is the graph where the, the rate of reaction increases and reaches to the maximum velocity. This is the velocity of the reaction. Okay, velocity of reaction. So when it reaches to the maximum, There is no further increase possible by adding of substrate molecule. After the reaction has reached the maximum, after the enzyme has bound completely with the substrate, if you keep on adding substrate, the reaction will not further get increased. That is what it means, because it has reached to the maximum velocity. This is called Vmax. Are you understanding? When a substrate concentration is increased, the rate of enzymatic reaction also increases first. Initially, they are in the race. Substrate is added, enzyme is added, they keep on, they increase to the rate of reaction. At a point, they reach to the maximum velocity. After this point, if you keep adding substrate, don't accept it to run further. No, there is no further increase. This is what it says. I write down this. Any doubts? Previous slide, this one, pH. Yeah. Can you hear me now? No, see, substrate is added, you add the enzyme molecules. After a while, all the places on the enzyme is being occupied by substrate. If you keep adding your substrate molecule, it will not reach to the after once the maximum velocity is reached, it will not further increases. Yash, is it clear? Can you see the screen?
Yes, ma'am.
Okay. So now this is a very important graph that you should know. See, V max is this is a initially this is a velocity of the reaction and this is a substrate concentration. S is a substrate concentration and V is a velocity of the reaction. Initially, what happened when the substrate is added, the enzyme molecules that binds and keep on increasing. All the enzyme molecules, the sites on the enzyme molecules is occupied by the substrate. After that, if we keep on increasing, it will not reach to any further increase because it has already reached to the maximum velocity. So this is the Vmax. Important to know. Next, what is half of Vmax? Vmax, half of Vmax is 50% of maximum velocity. When you extrapolate the 50% of maximum velocity on the x-axis, that will give you Km. I'll tell you what is Km. So Vmax is Vmax and 50 half of Vmax is 50% of the velocity of the reaction. When you extrapolate on the graph, you get Km. So what do you mean by Km? Please write down. Km is Michaelis constant, important MCQ. It's a mathematical deviation. Write down. Or it is a constant, which indicate, what does it indicate? Substrate concentration at which half the maximum velocity of enzyme reaction is attained. That is, Km is equal to Vmax by 2. Vmax is maximum velocity, right? Maximum velocity. So Km indicates half of Vmax. Are you all getting the point? Km indicates half of Vmax. If Vmax is 100%, Km will be is equal to 50%. So you can also write it as it is amount of substrate concentration to bring rate of reaction to half of its maximum velocity. That is Vmax by 2. That is Vmax by 2. We do not have to bring the velocity to half, but if we want to know the km, okay, then we have to see the half of Vmax, half of max velocity, maximum velocity. That will give you km. If you want to calculate the km value, we have to know Vmax, and half of Vmax will give you the value of km. How much substrate is being used to get a velocity for half. Then we know how much is for Vmax. Is it clear, Sanika? Yes. There's a bit derivation for the Km constant, which is not required for you all, but yes, it's a mathematical derivation. The different steps, how you get Km is equal to Vmax by 2, but you need to know this part, Km is equal to Vmax by 2. What is Km? Which constant is Km? And what does it gives? These are the three things that you should know about the Km. You do not have to know about the derivation. Is it clear?
Ma'am,
Yes.
could you please show me the slide of effects of pH?
Yeah, I'll show you the slide of effects of pH. Are you done with this? Michaelis constant.
Yes, ma'am.
Important. So what does it mean? It is an amount of substrate concentration to bring the rate of reaction to half of its maximum velocity. So when you know Vmax, you can calculate the Km, and Km is a substrate concentration where Vmax is equal to Vmax by 2. And this graph is important. Please copy this graph, a very important graph. Many times the MCQs are from this graph. Copy this graph. Done? So if you want to know the initial reaction rate or the velocity, that is the Michaelis equation, where you have V is equal to Vmax into substrate concentration, and Km plus substrate concentration will give you velocity of the reaction. Vmax is a maximum velocity, half Vmax is a 50% of the maximum velocity, and Km is a substrate concentration, at which Vmax is half. And yes, you know, it is a substrate concentration. So any doubts to anyone before we move to the next? Please go through it quickly. Okay, which slide do you want? Is this the slide you're talking about?
Ma'am,
Same
effects
to the
of
same
pH. The before one.
effects of pH. Okay,
Yes, ma'am,
rate
this
of
one.
almost. Yeah, you can take it down. Others quickly revise.
Thank you, ma'am.
You're welcome. Any doubts to anyone there? Anyone with doubts? Can we move to the next topic? The next topic is mechanism of enzyme action. Mechanism of enzyme action. The first theory that we're discussing about the hypothesis that we're discussing under the mechanism of enzyme action is lock and key hypothesis. Lock and key hypothesis. Important question. Who proposed or who gave this lock and key hypothesis is by Emil Fischer. In which year? In 1894. And this theory is based entirely on geometric shapes. Are you all there? This theory is based on geometric shape. Yes, everyone? Now, here, geometric shape, it means it's correspond to both enzyme as well as substrate. Yeah, enzyme and substrate, they both refer to lock and key. So a particular lock opens with a particular key. Do you agree with me? A particular lock opens with a particular key. Do you agree? Yes. So a particular lock opens with a particular key. Same way, a particular substrate can bind to a particular enzyme. Why? Because you know that they bind at active site. So for example, if this, let me get back to Word file. Just look on this. If you have an enzyme, if this is an enzyme, okay, if this is an enzyme, and to this enzyme, you're adding a substrate. Your substrate is something like this. A substrate, and you have B substrate. So tell me whether the A substrate will bind with the enzyme or the B substrate bind with the enzyme. I'm audible. Yeah, it is substrate A that's going to bind. Do you all agree with me? Substrate A that's going to bind. This is called the geometrical equations. It's between the geometrical shape. The binding is happening due to a geometrical shape, forming what? Enzyme, substrate, complex. Now, after that, you get a product. You get a product. This is a product. Do you think you'll get an enzyme? Do you get an enzyme? Do you think you get an enzyme? Yes. So that enzyme remains to be unchanged or it is changed. that enzyme is remained to be unchanged or changed? Unchanged. Very good. So you get your enzyme back. So you get your enzyme back. Is this point clear? Yes or no? So this is the equation for lock and key hypothesis. Who gave the lock and key hypothesis? Quickly tell me the answer. Who gave the lock and key hypothesis?
Emil Pischer.
Emil Fisher. Very nice. Have you understood this part about the
Excuse me.
lock and key? Yes, yes.
Ma'am, now the enzyme combines with the substrate. It converts the substrate into product, and again,
Yes.
enzyme is formed
Free.
means it is separated with the
Yes.
product.
Yes. You get your enzyme back, and this enzyme you can reuse for next reaction. Reuse for next
Okay,
reaction.
ma'am.
Okay.
Thank you.
You're welcome. Any doubts to anyone? Here, for example, enzyme will act like a lock and substrate will act like a key. That's the difference. Getting the point? Enzyme will act like a lock and substrate will act like a key. Next, can we move to the next topic? Have you all understood this part? Are you all there? Are you all there?
Ma'am, scroll up a bit.
Okay. Write it down.
Yes, ma'am. Thank you.
Induced fit hypothesis. Induced fit hypothesis. Induced fit hypothesis is proposed by Koshland. Proposed by Koshland. It's called Daniel Koshland in 1958. In this, remember, active site is not present in the enzyme. Active site for the substrate is not present in the enzyme initially. But once the enzyme is exposed to substrate, this causes the changes in the active site so that it allows the binding of substrate and enzyme. If you see, initially the active site is not created in the enzyme. Once you add a substrate, you know the substrate which is coming is something like this. This will change itself. This will change itself according to the shape of the substrate. That is what induced fit model says. That is what induced fit model says. Write down this point. Write down this point. I'll explain you
Ma'am,
again.
it happens in every case.
No, see, this is the hypothesis. Hypothesis means what? They are putting their view. The scientists are putting their view. Once one of the scientists said it is a lock and key mechanism, other scientists is saying it's an induced fit model. Are you getting the point? This is their thinking about the enzyme and substrate. This might be the way the reaction is taking place. That might be the way the reaction is taking place. Are you understanding? So that was the
Yes.
first one was lock and key mechanism, and now we are discussing the induced fit model. So in induced fit model, basically it says that here enzyme is not having any active site, but once a substrate, once it is exposed to substrate, then this enzyme can change or remold themselves to get into binding with the substrate. This is called induced fit model. Are you getting the point? Please write down. Sanjana, please write down this induced fit, then I'll show you the slide. Okay.
Ma'am, we should draw the diagram.
Yeah, you can draw the diagram. See, diagrams are important for your exams. You should know them. I always advise to, whenever you write a question, it's always support them with the diagram. That makes it easier for the examiner to understand what have you written in the paper. So always make an attempt of drawing the diagram as you get time. If they have asked you to explain, you have to write it compulsory. If they have not asked you, but yes, you can still, if you have get time, you can still explain them with the help of the diagram. This give a good impression. Yes, okay, I'll explain you. Listen to this, Yes. What do you understand by induced fit model? See, first thing is induced fit model is given by Daniel Koshland in 1958. What do you mean by this? It means enzyme will not have any kind of an active site to the substrate. But after it exposed to the substrate, enzyme will reframe its active site according to the substrate so that it can bind and give the product. Got the point? See, for example, if this is a, let me show you this. For example, if you see, this is a substrate, right? This is a binding site on the enzyme. This is an enzyme, but the substrate is something like this. Do you have a substrate binding site? No. After it is exposed to substrate, the enzyme will change its shape so that it allows the substrate to bind to form enzyme substrate complex. After it forms enzyme substrate complex, you have your product here, and then you have your enzyme which is unchanged. Please read this and write. It clearly says. It's not a, this is a hypothesis given by Koshland. It's no more specific. Yes, it's no more specific. As the enzyme, as a substrate comes, it will modify its active sites and then it binds. This is the hypothesis. It is not proved. It's just a said, proposed. Is it clear? Any doubts to anyone? Hello? Yes?
No, ma'am.
Okay, ma'am, I'm a bit busy. I'll call you later. Yeah. Okay. So now next, clear to all? According to this theory, write down, according to this theory, enzymes or active site is flexible. According to this, enzyme or active site is flexible. One is you have buttering site. It has two groups, okay? Active sites of enzyme contain two groups. Active site of enzyme contains two groups. One is buttressing group. Other is catalytic group. Buttressing group is meant for supporting the substrate or the attachment of substrate. Buttressing group is for attaching the substrate. Whereas catalytic group is for the catalyzes the reaction, for catalyzing the reactions, to bringing out the reaction, catalyzing the reaction. Okay, now let us draw this with the diagram, it will be more clear. So you have an enzyme. This is your enzyme. Enzyme is having one side is catalytic site, C is catalytic site, and whereas this side is B is buttressing site. Plus there is a substrate molecule. Plus there is a substrate. Okay. Now what happened if this to this enzyme substrates comes and bind. Substrate bind to this enzyme. Then there is a conformational changes in the enzyme. Conformational changes in the enzyme. The conformational changes in the enzyme is in such a way that the catalytic and the substrate will come closer. Something like this. They bend, and here in the middle there is substrate binding. Here you have buttressing site. Here you have what? Catalytic site. So the changes is in such a way that you get the catalytic sites comes towards the substrate, forming a enzyme substrate complex or enzyme product complex. Then you get your enzyme back. After the reaction. You get your enzyme back, and also you get your product back. You get your product back. Plus you get your product. Did you understood how does it work? Initially, the enzyme will not have what specificity. But once the enzyme is exposed to the substrate, it changes its conformation in such a way that it binds and bring out the reaction and result in the formation of product. Clear? How many times the connection is going, Yash? Okay, from where? From here? Right down. What about others? Are you understanding? Zenobia, Shruti, Vaibhavi, Sakshi, Rej, Raghavi, Madhura, Sneha, Desai, Dared. Are you understanding? Sam? Yeah, Sanjana, I'll show you. Just 2 minutes. After this, you remind me, I'll show you. Yes, are you done with this according to the theory? Or should I move down? Should I move it down? Is it Meher is from this class?
Ma'am, she's for coaching, I guess.
Coaching. Okay, fine. Can I show the next point? Okay, you can just write down or copy down this quickly.
Ma'am,
Yeah.
can you explain this process again?
Who's this? Tanya.
Yes, ma'am.
Okay, listen. Listen to this. See, in this induced fit model, here enzyme will not have an active site for substrate. You know active site. Active site is that site to which enzyme and substrate bind. But in this model, there is no active site which is present for the substrate. After you expose the enzyme to the substrate, active site is formed. After it is exposed to the substrate, active site is formed. And then the substrate will give rise to the reaction will take place and the formation of product. For example, you have two sites in the enzyme. One is catalytic site, other is buttering site. Then the enzyme, then we add substrate. What the substrate will do? Substrate will go and bind to buttering site. After it bind to buttering site, there is a conformational change that takes place in the enzyme in such a way that catalytic site and the buttering site will come together and bring about the reaction with the formation of product and unchanged enzyme, because enzyme always remain to be unchanged. Is it point clear?
Yes,
Others, any doubts until here?
No, ma'am.
Okay. Okay, Sanjana, which slide do you want to show? Lock and key, right? Okay, write down this lock and key. Is this a slide you're asking for, Sanjana? Okay, take a screenshot. Okay, fine, done. So this is all about induced fit theory. Now everybody please go through this lock and key and induced fit theory before we move to the next. Okay. Now we'll be studying about the inhibitors. Okay, inhibitors. Inhibitors, what are inhibitors? Write down inhibitors. Inhibitors are the substances which prevent substrate to convert into product, which prevents substrate to convert into product by binding with enzyme. What do you understand by this? See, you have enzyme, you have substrate. If substrate binds with enzyme, then you get product, right? So this inhibitors, I is inhibitor. Inhibitors prevents this binding so that there should not be any formation of product. So are the substances which prevent substrate to convert into product by binding with enzyme. They go and bind with enzyme, but they do not allow a substrate to bind so that they do not form any product. Did you all understood what is inhibitor? Did you all understood what is inhibitor? Where inhibitors are the substrates which prevent the substances which prevent the substrate to convert into product by binding with enzymes. They bind with enzyme. They'll not allow the enzyme to bind with substrate. So means you're going and occupying that seat where substrate should come and sit so that the product can be formed. But what they do, what inhibitors do, they go and occupy the seat so that there should not be any formation of product. Did you got the point, everyone in the class? Everyone in the class.
Next. Yes, it prevents the whole substrate to convert into. Okay, how it's going to help? We'll study with the example, Sanjana. Okay, we'll study with the example now. But yes, it will not allow the product to form. Here you have two types of inhibitors. One is competitive inhibition, competitive inhibition, and the other is non-competitive. One is competitive, other is non-competitive. So first let us discuss about the competitive. Are you all ready? Are you all there? Active in the class. Competitive means yes and I. The competition is between the yes and I. The competition is between the S and I to bind to enzyme. How a competition? Here, yes and I have almost similar structure. Is it clear? S and I have almost similar structure. Are you understanding? So when you have similar structure and when there is one seed, for example, you have an enzyme. You have an enzyme. This enzyme is having a structure like this. This is an active site. This is an active site. This is the substrate. And also you have a inhibitor which is same. So don't you think they both will compete to bind to this site? Tell me. Don't you think this both will compete to bind to the active site because they both are of similar structure? Yes. So that's a competition between a substrate and enzyme. That's a competition between substrate and inhibitor to bind to enzyme. Did you got the point? What is competitive inhibition? Here, they compete to bind. Here, they compete to bind with active site on enzyme. Okay. Now, very important. This reaction is reversible. When? It's reversible, but when? Reversible on increasing substrate concentration. When is it reversible? When on increasing, it's reversible. The reaction can be reversed. Only substrate can bind. When you have more of substrate concentration, when you have more of yes everywhere and less of I, you can get a product because there are more of substrate concentration. Is this point clear? Let us draw this graph. The same graph. With inhibitor without inhibitor. Then you get, so what is this? Tell me, what is this? Okay, Sanika, I'll scroll up. Tell me what is this on the y-axis? What is this? Quickly. Nobody can answer this? Simple. Very good, Yash. It is Vmax. How Km, Sanjana? Km is on the substrate concentration. Km should be on x-axis, not on y-axis. On y-axis, we are talking about the rate of reaction in velocity. Sanjana, are you getting the point? Still, if you have doubts, please stop me. Velocity is on y-axis. Is it clear? Okay. Now, this is Vmax. Now, what is half of Vmax? This is 50%. Okay, half of Vmax. So when you extrapolate it, here you get, what is this? What is this? Very good. That is now Km. That is now Km. This is Km, 1 Km, and this is other Km, right? You get two different Km, but what about the Vmax? Vmax remains to be changed. So if the question is asked, what happened to Vmax in competitive inhibition? What will be your answer? Vmax remain changed or unchanged in competitive inhibition? Remain unchanged. Do you all agree with this sentence? Remains unchanged. Very good. Whereas Km value is increased. Value is increased. Whereas Km value is increased. Clear? Any doubts? See, I'll revise it again. Competitive inhibition is where your S and I almost have similar structure. So when they have similar structure, there is a competition between I and S. I is an inhibitor and S is a substrate. When why do they compete? They compete so that they can bind to the active site on the enzyme. This reaction, if the inhibitor binds, then there is no product. If the substrate binds to the enzyme, there is a product. This reaction can be reversible if you increase the amount of substrate concentration. So you get a graph where you have with inhibitor and without inhibitor. With or without inhibitor, in this graph, your Vmax remains to be changed, but the Km, there is a difference where the Km value increases with inhibitor. Why? Because more of substrate you're adding more substrate. To get that 50%, you require more substrate. So the Km value increases. Very important question, very important MCQ. This is both questions. Very important MCQ. Very important MCQ. Graph is important. Did you all understood? It's important with respect to your main exam as well as for your medical exam. Any doubts to anyone? Go through it. Take your time. Clear? Any doubts? Any doubts to anyone? Yeah. Then my idea. Any doubts? Mod, Sanjana? Yes. Sam? Sania? Yes, this is the third time you're losing in the connection. Yeah. Okay, let me explain you again. Can you hear me now, Yash? Which part you want me to explain? Others, please revise. Okay. Are you, which part you want me to explain? Competitive inhibition. Second part of active site. Which is that? Can you just tell me which is that? Second part of active site. What do you mean by that? Two parts of active site. Acha, acha. Two parts of active site. Okay. See, listen. Others, please revise. The active sites parts have two groups. One is buttressing group, other is catalytic group. Buttressing group is for attaching the substrate, and catalytic group is to bring the reaction, catalytic reaction that convert the substrate into product. So you have buttressing group, you have catalytic group. Buttressing group to which the substrate molecules and goes and bind, and this bring about the conformational change in the enzyme. Okay, to the buttressing group, substrate will bind, and after that there's a conformational change that takes place. The conformational change happens in such a way that the catalytic site comes closer to the buttressing site, and then this forms the product and unchanged enzyme. Yes, clear? Okay. Any other question or query for today's class? Any other question or query for today's class? So each and everyone in the class have understood these concepts. That's okay, no problem, Ish. Okay, let me ask you a few questions. Okay. Have you all revised? Can I ask you a few questions? Who proposed the induced fit hypothesis? First question. Induced fit hypothesis. Koshland. Good. Daniel. Okay, good. Next question. Does enzyme have does the active site present on the enzyme is similar to the substrate in induced fit hypothesis model? Very good. No. Very good. Km is dash constant. What is the name of the constant? Michaelis constant. Very nice. Next is which are the two groups that are present on active site? Which are the two groups that are present on active site? catalytic and buttressing site. To which of this group that does the substrate bind? To which of this group does the substrate bind? To which of this group that substrate, Sakshi substrate. Sanjana, good. It's a buttressing site. The substrate will bind. That will result in the change in the conformation. Is it clear? Okay. Lock and key hypothesis is based on what? Lock and key hypothesis is based on what? Geometrical shapes. Very nice. Any doubts for today's class? Okay. Any doubts for today's class? Okay. So I'll be logging out. You're welcome. Bye bye. Take care. Welcome. Bye bye.